that I've been working on for the past 6 years. I'm really excited to share this work with you, but before I get into the specifics of my research, I want to give you a little background in my field of study. I work in population dynamics, which essentially is a field that uses math and equations to represent population sizes of organisms. Now, this field started with single species models, where you have one equation that represents one population size of a single organism, and you're using that math to both estimate the population size now and predict potential sizes in the future. These models were added to in complexity with the creation of predator-prey dynamic models, where you include both the population size of your species of interest and their prey resource, since that will have a big impact on their potential future population sizes. Further additions were made in competitive models, where other predators who consume the same prey resource were also included, as they can both directly and indirectly affect the population size of your species of interest. From here, we expanded all the way to food web models, where all organisms living within a food web that can either directly or indirectly affect your population of interest are included. Now, food web models were even further expanded upon with the creation of ecosystem-based models, which take your food web and combine it with different outside components of the ecosystem and environmental drivers to improve your predictions. Single species models are still in use today, and they're still very important in accurate predictions of single species dynamics. However, the global call has been to move towards ecosystem-based fisheries management, and ecosystem-based models are a useful tool in helping manage the system from a holistic view that produces healthy ecosystems to provide the most resources possible. However, when you are looking at your ecosystem, an event like this occurs. This is the Deepwater Horizon oil rig explosion that occurred in April 2010, resulting in 11 deaths and oil that spilled into the Gulf of Mexico for about 3 years. And when you have an event like this and you see organisms in your system that look like this, or like this, or like this, the question arises, what impact is this having on your ecosystem as a whole? These fishes have clear external oil impacts, but what does that mean for their individual health, the health of the population, and the health of your ecosystem? These questions were the driver for the goal of my dissertation: incorporate oil spill effects into Atlantis, an ecosystem-based model. Now, I chose Atlantis because it's a comprehensive modeling tool that combines our food web with different subroutines of the system, like physics and chemistry of the water, as well as dynamic fishing fleets, to capture the important drivers affecting our fishes. Now, Atlantis is a computer simulation software, but I'm going to give you a visual representation of what it's doing with this box. So for now, let's pretend that this box lives on my computer and it's filled with water. Inside this three-dimensional box, there are nutrients and detritus, as well as all of the organisms in our food web. These organisms are represented in populations that Atlantis calls functional groups. Functional groups can be as specific as a single species that you are interested in, or a group of similar species like bivalves. Within this box, all of these components are distributed evenly through space, or homogeneously. There is no aggregating or clumping like you see on the right now. Now, depending on the region of the ocean that you're representing with your box, you can divide it up to 6 depth layers, sort of like a layered cake. There is also a complementary sediment layer at the bottom of every box. These layers interact with each other through the movement of water, nutrients, and organisms. There are dynamic fishing fleets that move across the surface of these boxes to target their species of interest. Now, throughout this slide, I've been saying box, but actually, Atlantis is not limited to a box structure. It uses a polygon structure. So let's take an aerial view of our previous box, and it looks like this. But it could, in theory, look like any of these shapes. A polygon is any shape that has completely straight edges. So if you take 66 of these polygons and you overlay them in the Gulf of Mexico, you have our Gulf of Mexico Atlantis model, shown here in a map with all 66 polygons that have up to 6 depth layers, depending on the region they represent, and a separate sediment layer. These polygons, along with their depth layers interacting vertically, can interact with their neighbors through horizontal movement. Now the question is, when you have this polygon structure, what happens when you introduce a deep sea oil spill and oil pervades throughout your system? This was the genesis of my dissertation goal to incorporate oil spill effects into Atlantis, which was driven by three main questions. They are: question 1, how does an oil spill affect fishes and how can we model it? Question 2, how does an oil spill affect invertebrates and how can we model it? And question 3, how does the affected Atlantis ecosystem respond over time? Now, questions 1 and 2 were done to define the relationship between organisms and oil exposure so that those relationships could be added to Atlantis in question 3 and simulations over time conducted to test possible long-term impacts. For now, we'll start with question 1. To address this question, I first decided to look at what is already known in the literature about the relationship between fishes and oil exposure. Here is an excerpt from a table dating back to 1974 showing a summary of different marine organisms and the estimated range of oil concentration causing toxicity, and in this case, toxicity means mortality. If we're focusing on fishes, since that's the target of our question, you can see that the range causing mortality is 5 to 50 parts per million. However, for the purposes of dynamic, spatial, and temporal ecosystem modeling, this is kind of a big range to try and decide where should mortality start. And also, is mortality the only effect that we would see in our populations from oil exposure? What I really needed to know was, as oil varies across any range of exposure values, what is the population impact? And not just mortality, what population dynamics are affected by this oil exposure? So I actually looked at 3 population dynamics, and they are growth, mortality, and recruitment. For now, we're going to focus on growth and mortality. So I need to define, for both growth and mortality, what is the relative impact to those dynamics as oil exposures vary. To answer this question, I collected available data on growth and mortality impacts on fish exposure from oil. At the time of this study, there was not a lot of data available directly from the Gulf of Mexico, so I used any published data set relating to oil exposure and fishes. I took these data sets and I fit 4 potential dose-response models to them, describing the relationship between oil exposure and population impact. They are a linear model, which indicates that as oil exposure increases, population impacts increase linearly. A hockey stick model, which suggests that there is a threshold below which you do not see population impacts from oil exposure. But when that threshold is passed, population level impacts occur and increase. A stepwise model, which has the same threshold concept as our hockey stick, but instead of an increasing impact after that threshold is passed, you have a constant population impact above a certain threshold. And lastly, an exponential model, which suggests that as oil exposure increases, population impacts increase exponentially. Starting with growth, here's the data set that I used to assess these 4 models. On the y-axis, you have the growth reduction proportion, and on the x-axis is the oil exposure value. Please keep in mind that the x-axes I will be showing you are in a log scale. You may also note that there are 3 different exposure types shown on this graph. Fish in these studies were exposed either via food, sediment, or water. So the first question I had to address before I could fit my 4 models was, does exposure type matter? While we know that the route of chemical ingestion through these different exposure mediums is different in the body of fishes, the question for me was, does this have a statistically significant impact on the population level outcome? So I tested the 3 exposure mediums, and for growth, I actually determined that while water and sediment had no significant differences in outcome, food was significantly different and had to be tested separately. Here is the sediment and water data set with my 4 dose-response models fit. They were fit using linear and nonlinear least squared residuals, which essentially means I tried to minimize the distance between each model and all of the data points in my data set. They were then assessed for parsimony using AIC. And parsimony is a cost-benefit analysis that essentially asks, is it worthwhile to have a more complicated model that fits your data points better, or is the better option keeping a simpler model that actually doesn't fit quite as well? Using both of these methods, this data set, I determined that the hockey stick dose-response model was the best fit and most parsimonious in this case, suggesting that there is a threshold below which impacts are not seen. I used the same methodology for my food exposure data set for growth, and the same tests once again indicated that the hockey stick model was both the best fit and most parsimonious. Moving on to mortality, I want to take a step back and remind everyone of these images. While direct mortality from very high concentrations of oil on adult fishes occurs, we see significant sublethal impacts at lower oil concentrations. And it is established that fishes with a reduced state of health are more susceptible to mortality from other sources, such as predation, starvation, competition, and disease. So instead of limiting our data set to only direct mortality, I actually used a reduced health indicator data set, things like tumors, lesions, and fin rot, as a proxy for an increased likelihood of mortality in Atlantis. And that is the data set that you see here. I ran the same exposure medium test on this data set to determine, did exposure medium have a significant effect? And for this health indicator data set, it did not, meaning I could keep all 3 exposure mediums contained in one larger data set. I fit my 4 models to this data, and using the same fit and parsimony tests, shockingly, once again, the hockey stick model was selected as the best fit and most parsimonious, really driving home that there is a threshold below which population impacts on adult fishes from oil exposure are not seen. Lastly, for recruitment, I was lucky enough to have a colleague in the Sea Image Consortium doing her master's thesis on recruitment impacts from the Deepwater Horizon oil spill. So instead of conducting our own study, we used Emily Chancellor's master's work and implemented it into Atlantis. And I will give you a brief summary of her work here. She calculated the proportion of annual production of fish larvae potentially exposed to Deepwater Horizon oil. To do so, she took the abundance of larvae at the surface within the oil spill area from months April to July, when surface oil was present. She divided this abundance by the abundance within her entire sample area for the entire year. Or in other words, Emily calculated the percent of all larvae in the Gulf of Mexico that were in the wrong place at the wrong time. She took these percents and applied direct mortality to all of the larvae encountering surface oil in Atlantis. In summary, for question 1, I found that exposure medium was significant for growth effects and therefore had to be tested separately, but not for mortality or proxy mortality data. All data sets indicated that the hockey stick shape is both the best fit and most parsimonious, which confirms what we saw in that previous table, that there is a threshold below which fishes are not affected, and then after that threshold you see population level effects. For question 2, how does an oil spill affect invertebrates and how can we model it? I started with the same conceptual map that we saw in question 1. As oil varies, what is the impact on invertebrates? And specifically here, I looked at invertebrate abundance. There's a previously described relationship between invertebrate abundance and oil in the literature. And it suggests that at low oil concentrations, you actually see an increase in abundance. And then as oil concentrations increase, abundance declines again. So first, I wanted to test, was this relationship present in the Gulf of Mexico after the Deepwater Horizon oil spill? I have 2 data sets available to me, one for macrofauna abundance, which is a term that you usually associate with the word invertebrate, things like shrimps, worms, and bivalves, and a microfauna data set, which is less known to the layman anyway, and usually represents smaller organisms living inside the sediment. This data was provided by Paul Montaña's group at Texas A&M University and were collected from sediment samples in September and October of 2010, so 5 to 6 months after the oil spill started. But before I could determine did this relationship exist in those 2 data sets, I needed to make sure that it was the best descriptor of the relationship between abundance and oil exposure. So I fit this domed shape relationship or polynomial model along with 3 other ecologically relevant models: an exponential relationship between invertebrate abundance and oil, a linear relationship between invertebrate abundance and oil, and a constant or no relationship between invertebrate abundance and oil exposure. And here those 4 models fit to the macrofauna and myofauna data sets. I've zoomed in quite a bit on the y-axis for macrofauna just to try and show you the different fits of those 2 models, but there is quite a bit of overlap between linear and constant. I assessed these 4 models using the same methods seen in question 1, tests for fit and tests for parsimony. And for both data sets, for both fit and parsimony, the polynomial or domed relationship was selected as the best fit and most parsimonious. Hooray! So now I've confirmed this relationship existed in the Gulf of Mexico, which is great news for me. But how do I drive this relationship in Atlantis? I have that previously defined hockey stick dynamic, which can induce toxicity once a certain oil threshold is passed. But what process is driving increases in abundance at low oil concentrations? And can I implement that ecological process in Atlantis? To determine this, I ran 3 possible test scenarios that could describe an ecological process increasing invertebrate abundance. And I will list them for you here and then go into more detail on. Scenario 1 is extra nutrients from Mississippi flushing. Scenario 2, extra detritus from surface oil aggregation. And scenario 3, predators avoid oiled regions. So to start with scenario 1, this is a satellite image of the Gulf of Mexico. And in this red box is the Mississippi Delta outflow region. Now during the Deepwater Horizon oil spill, in an attempt to push oil away from coastlines, the U.S. flushed the Mississippi River, which essentially means its flow rate. And the Mississippi is an important source of sediments and nutrients in the Gulf of Mexico. So with increased flow rate comes increased nutrients. With these nutrients, primary producers could potentially grow in population size, which adds food to the system. You may be more familiar with this process from this image of an algal bloom. It's the same concept. When you add nutrients to a system, it provides food for the planktonic photosynthesizers at the surface, which in turn can provide food for the food web as a whole. Scenario 2 is extra detritus from surface oil aggregation. I will do my best to provide you with a very simplistic overview of particulate dynamics at the surface of the ocean here and how oil interacts with those dynamics. So in my box ocean at the surface, we have 2 types of particulate matter, organic and inorganic. Organic matter are things like detritus and fecal pellets. Inorganic matter are things like minerals and sediments. Now these particles sink through the water column at a rate dependent upon their size. And as they sink, they are susceptible, some of them, to consumption from microbes. This consumption in turn makes them smaller, and they sink slower, and it's a vicious cycle until some particles never reach the bottom of the ocean before they are either fully consumed or buoyant at depth. Now when you add oil to this system at the surface, oil interacts with other components of the surface waters and actually forms a sticky glue that creates large aggregates of particulate matter. These larger aggregates weigh more and therefore sink faster. And because they're sinking faster, there is less time in the water column for consumption to occur, overall resulting in more organic matter reaching the bottom of the ocean. This organic matter can be food for certain components of the benthic community, which could potentially increase in invertebrate abundance. Now this process that I've described has been named marine oil snow sedimentation and flocculant accumulation, or MOSFA. So when I'm referring to MOSFA in the future, it's this oil sticky glue rapid sinking that I'm talking about. Lastly, for scenario 3, it has been shown that some fishes, when exposed to contaminated water, will actually move to sources of cleaner water when available. Specifically to the Gulf of Mexico, this could mean that if a certain region was exposed to oil, the fish predators would then leave that area, releasing the invertebrate populations from predation pressure and allowing their population sizes to grow. These three scenarios, I first needed to test on their own, without the presence of toxicity, are they able to induce the first half of this dome, or an increase in invertebrate abundance? So I tested them by themselves, and really quickly, here are the three scenarios again, and these are the results. On our y-axis, we have the average invertebrate abundance increase when compared to a control Atlantis simulation with no environmental drivers. You can see that all three scenarios did induce an increase in invertebrate abundance compared to the control. However, scenario 2 had a significantly larger impact on the relative increase. But all three scenarios technically did their job. I combined them with toxicity for all three and ran those to determine did both halves of the domed relationship occur. And here are those results. These are polygon maps of our Atlantis Gulf of Mexico model, and a polygon that is shaded red has a net decrease in invertebrate abundance when compared to a control scenario, and a polygon that is shaded blue has a net increase in invertebrate abundance when compared to a control. You may notice that there are no blue polygons in scenarios 1 and 3. And actually, in these two scenarios, any potential invertebrate abundance increases were overwhelmed by the application of toxicity. And only scenario 2 indicated both halves of our domed relationship at the polygon level. Now, this is aggregated across all invertebrate functional groups, and I wanted to dive a little deeper into scenario 2 and determine what is happening at the functional group level. So these graphs, before I show you the results, to give you a little orientation, similar to previous graphs, as you move right along the x-axis, we are increasing in cumulative oil exposure. And as you move up or down on the y-axis, it's either an increase or decrease in abundance compared to our control. And here are the results for 9 functional groups. You may notice that for some of them, there is a domed relationship. For some functional groups, this relationship is stronger on one side or the other, but it suggests that the combination of MOSFA and toxicity in Atlantis, both at the invertebrate functional group aggregation and at functional group individually, we are able to drive the domed relationship. You may notice that there are actually 2 groups showing an inverse domed relationship. Since this was just a test of efficacy, or did this system work for invertebrates as a whole, we're not actually concerned with the fact that these 2 groups are doing the inverse. But it is likely because of food web dynamics. Since all invertebrate functional groups had the same toxicity applied and the same amount of detritus added to the system, there has to be an inherent dynamic in our food web model driving these inverse relationships. In summary, sediment cores from the Gulf of Mexico indicated the domed relationship occurred in September and October of 2010 after the Deepwater Horizon oil spill. All 3 scenarios tested independently induced increases in abundance. However, only scenario 2, the detritus from MOSFA, was able to drive both increases and decreases in abundance across oil exposure. Finally, for question 3, how does the affected Atlantis ecosystem respond over time? I took the results from question 1 and 2 and fed them into 50-year simulations in Atlantis. Specifically, I used our hockey stick dose-response model of mortality on both invertebrates and fishes and growth for fishes, combined with Emily Chancellor's estimates of larval mortality, the detritus forcings from MOSFA, and lastly, dynamic emergency fisheries response closures that were done in response to the Deepwater Horizon oil spill to mitigate impacts. Now, to drive oil dynamics in Atlantis, we actually received a far-field oil model output from Claire Harris's group at the University of Miami from her connectivity modeling system. Claire provided 167 days of oil outputs at 1/12th degree latitude-longitude grid points. However, if you'll remember from our previous talk of polygon dynamics, there is no spatial aggregation inside a polygon. So we took Claire's grid points and averaged them across the area of the polygon and depth layer within which they fell. And this visual is actually the cumulative oil for all 167 days, and it's also summed across depths, just so that you can visually see the polygons that were most impacted. But for simulation purposes, we did not do cumulative days and cumulative depths. For the model results that I'm going to show you, I'll be highlighting impacts from the 13 most affected polygons. That way the signal is visible and not lost as if you had averaged across all 66 polygons, which may or may not have been affected. I will also be aggregating functional groups into guilds. So taking a step back, if you remember, functional groups can be things like specific species or similar groups of species. Guilds are essentially groups of groups, or similar functional groups aggregated into one larger group. Here are the first visuals of our simulation results for our 50-year runs. On the y-axis is biomass relative to no oil spill, and on the x-axis is year. The solid blue line is the average biomass for all sensitivity analyses, and the shaded green region is the range of outcomes as we varied sensitivity of the system to toxicity and sediment oil. You'll notice in both of these guilds that there is an initial decline right around 2010 when the oil spill occurs, and then depending on your sensitivity range, some recovery, either completely recovering to the no oil population size or stabilizing at a slightly reduced population size throughout the duration of the simulation. Now this is in contrast to our two most impacted guilds in the system, large demersal fishes and elasmobranchs. Depending upon our sensitivity parameters, you can see the minimum that occurred during the oil spill time ranged from 25 to 50% of the no oil population size. And then recovery happens somewhat quickly, sometimes recovering completely, or in the case of elasmobranchs, stabilizing slightly below our no oil spill population size. Now these figures have been very specific to guild dynamics, but I also wanted to take a look at the ecosystem as a whole. So I'll be showing you two figures from two separate scenarios. First is the best case scenario, or the top of each green shaded region, when sensitivity parameters are weakest in terms of oil toxicity. Second is the worst case scenario, or the bottom of our green shaded region, when oil had the most impact. In these two scenarios, I wanted to determine, is there a relationship between this low point, or the biomass minimum that a guild sees, and the amount of time that it takes for them to recover or return to no oil spill population sizes, or in the case of elasmobranchs, no recovery. So here is the first figure for the best case sensitivity results. Each individual point is actually a functional group, but they are labeled at the guild level. On the y-axis, we have that biomass minimum, or how low did the population size go compared to our control. And on the x-axis, we have recovery time. Anything to the right of that 50-year dotted line did not recover in our Atlantis simulations, since we ended at 50 years. The first thing you may notice is that it is a shotgun pattern, or there is no clear relationship between the x and the y-axis. But there is still an interesting story here. In this best case scenario, the majority of our functional groups are recovering in the first 20 years. And then after 20 years, you have some groups recovering in the 20 to 50 year range, and then a second group that does not recover. Of those that do not recover, the majority of them have biomass minimum ranging from 70 to 95% of the no oil control population size. What this suggests is that in this scenario, the majority of functional groups that do not recover have dynamics similar to what we saw in snappers, an initial population decline ranging from 70 to 95%, and some recovery, but never to the pre-oil spill or no oil control values, stabilizing instead at a slightly reduced population size. Now this is in contrast to what we see in the worst case scenario. First note here is, again, there is still no relationship between the x and the y-axis. It's still a shotgun pattern, but it's different with a very different story to tell. You do not see a majority of functional groups recovering in the first 20 years in the worst case sensitivity results. And in fact, over half of functional groups in this scenario do not recover. And of those that do not recover, they have a wide range of biomass minimum. Some go extinct, some as low as 10%, up to that 90% range, suggesting that there is a wider range of population size impacts for these 13 polygons. Now, the contrast between these two figures is highlighting the importance of our understanding of both the oil field in the water column and the sediment, as well as the sensitivity of fishes to oil. Our dose-response model, or that hockey stick model, was created based off of a general fish response, but it's probable that different fishes would be impacted differently, and therefore improving our understanding of that hockey stick shape for different lifestyle of fishes would help us determine where in this best case, worst case range the Gulf of Mexico actually falls. Lastly, I want to look at a system already sensitive based on oil exposure in our worst case scenario and combine it with different fishing mortality values to see how does fishing effort affect our dynamics. So I took that worst case oil scenario and I combined it with F decreased by a factor of 2, or F times 0.5. F increased by a factor of 2, or F times 2, and F increased by a factor of 10, or F times 10. And I still maintained those emergency fisheries closures in these scenarios. Here are the results for the Snappers Guild. That light blue line in the middle is our base scenario that we've seen from previous graphs. You may notice that when fishing pressure is released, unsurprisingly, populations recover to a larger population size. But actually, this makes the difference in the worst case oil scenario between population recovery to a no-spill population size or not. And in our F times 2 and F times 10 scenarios, you can see there are significant population decreases. What's really interesting here is the magnitude of the effect of the oil spill at the very beginning varies based on our fishing pressure. And what really appears to be determining population size over time is not the fact that the oil spill occurred, but instead the fishing pressure used in the system. We see similar dynamics for groupers. While the effect of the oil spill has less of a range at the beginning, our long-term population sizes are still being driven by our fishing mortality and not by the initial decline in biomass seen from the oil spill. For shrimps, we see a little bit of a different story. All shrimps show severe biomass declines almost to extinction, but the recovery is very different for our 4 fishing scenarios. 3 of the 4 fishing scenarios, or all but F times 10, have recovery above no spill population sizes. What this is suggesting is that while clearly the oil spill had an effect initially, and fishing pressure is determining overall population size, there is some other dynamic occurring in the model that's allowing shrimp populations to grow larger over time than they would have if the oil spill had never occurred. Now, these figures, again, have been very specific to guild dynamics, and I wanted to look at the system as a whole. So I calculated the relative magnitude of impact for each guild, or in other words, the distance between our base fishing scenario and each individual fishing scenario for all 9 guilds. And that is what you see here. On the far left is F times 10, and clearly the most impacted group is snappers. As you move to the middle is F times 2, and snappers and groupers are roughly equally affected. And on the far right is F times 1/2. And you'll actually see that groupers show more biomass increase than snappers do. What this suggests is that snappers are more sensitive to the combination of intense toxicity of oil and fishing pressure and show greater declines. And when that toxicity to oil still occurs, but fishing pressure is released, they are less able to rebound in population size than groupers are. In summary, for question 3, Atlantis indicates that large demersal fishes, including elasmobranchs, were the most heavily impacted by the Deepwater Horizon oil spill. Our range of sensitivity analyses tell very different stories, highlighting the importance of understanding our toxicity dynamics for different types of fishes. And changes in fishing mortality can have a large effect on both biomass and recovery. Overall, towards the goal of my dissertation, I generated a general dose-response model that can be applied in any modeling scenario, that hockey stick model, and can be fit to any population impact data set. I confirmed the domed relationship for invertebrates was present in the Gulf of Mexico after the Deepwater Horizon oil spill occurred. I identified that MOSFA was an important model driver of this relationship and can be used in Atlantis to drive the domed shape. And lastly, Atlantis suggests that complete recovery within 50 years may not happen, depending on our sensitivity parameters, and that changes in fishing mortality can either compound or mitigate impacts. These are very specific to my application of the oil spill, and I wanted to leave you with information on global oil spills. However, to my knowledge, the number of global oil spills that have occurred since Deepwater Horizon is not a publicly available statistic. If anyone has that number, I'd love to hear it. I did check around. No one knew. But in my searching, I did find something interesting. I stumbled upon the incident news site for the National Oceanic and Atmospheric Administration, or NOAA, which is a U.S. federal governing body. So everything you hear here is specific to U.S. waters. Now this site tracks the number of oil and chemical-related incidents that NOAA has to respond to in U.S. waters. And NOAA defines something as response-worthy based on this. If it is a larger, more complex incident in which scientific expertise is required to track or clean up oil, or cases with a significant threat to marine and coastal resources. So this is not a total of oil spills and chemical-related incidents, it's only significant impact ones. And to give you a point of reference, this bar is when the Deepwater Horizon occurred. So keep in mind the size of the bar is not scaled to magnitude of event, it's just a count. And everything to the right of this bar is the number of incidents NOAA has responded to since 2010. Now this is across the US and is not oil specific, but this graph is actually an infographic on their website. So I was able to drill a little deeper and find out how many oil-related events have occurred in the Gulf of Mexico since 2010. And that number is 32. There have been 32 oil-related incidents that NOAA has had to respond to in the Gulf of Mexico since the Deepwater Horizon oil spill. And another quote directly from NOAA's website is that the Gulf of Mexico, a region which produces and refines a lot of oil, also experiences the most oil spill responses NOAA is involved with of any other US region. So what I'm getting at is that oil spills, especially in the Gulf of Mexico, are a constant, prevalent part of our system and are not going anywhere as oil and gas exploration continues. As we move towards ecosystem-based fisheries management, our ability to both incorporate these impacts into our population models and understand their long-term implications is going to be critical. So with that, I would like to thank my advisor, Dr. Cameron Ainsworth, for your support throughout my degree, my committee members and colleagues for your mentorship and guidance, my friends and family for always being there for me, and then one particular individual who really got me through my degree and I think had a similar impact on several other CMS students, my dog Hercules, who was somewhat of a local celebrity and I think will be sorely missed by the CMS student body. So with that, thank you again, and I'd like to open the floor for questions.